Welcome again friends, to a video in Beginners Bioinformatics series. Today we will try to understand how basic local alignment search tool or BLAST software works, its subtypes, and we will also use a demonstration for BLASTn, which is used for nucleotide queries. We will learn its important features and result analysis. As we know that the DNA bases are same in all living organisms, and the genetic codes for protein translation are also universal. This makes the nucleotide and protein sequences an important resource which can be used and compared among living organisms by aligning them. This will help to find similarities and differences in their genetic information, which can be further analyzed to find homologies and evolutionary relatedness among them. Basic Local Alignment Search Tool or BLAST is a software or tool which finds regions of local similarity between sequences by aligning and comparing them. It's a registered trademark of National Library of Medicine or NLM. BLAST generates mathematical scores and calculates the statistical significance of matches as e-values. Therefore, BLAST can be used to perform a fundamental operation of bioinformatics, that is to infer functional and evolutionary relationships between sequences and organisms, as well as help to identify members of gene families. At BLAST homepage, we will see Web BLAST, which has 5 types: nucleotide BLAST or BLASTn, protein BLAST or BLASTp, BLASTx, T-BLASTn and T-BLASTx, which is not indicated on the home page but is available for BLAST analysis. Upon scrolling down, we will see some more types of BLAST meant for specialized searches. This table explains various types of Web BLAST and where we can use them depending on our query type and the output we want. For example, in nucleotide BLAST, query has to be a nucleotide sequence, and database searched will be again a nucleotide database, and output will be nucleotide. Similarly, in protein, the query and the database will be protein and output will be also a protein sequence. Whereas in mixed BLAST, BLASTx uses a translated nucleotide query and searches against a protein database and gives us results as protein sequences. T-BLASTn uses a protein sequence as a query, searches against a translated nucleotide database, and the output is nucleotide. T-BLASTx uses a translated nucleotide query, searches against a nucleotide database translated in 6 open reading frames, and output is nucleotide. In today's tutorial, we will focus on most common types of BLAST web searches. Let's begin by clicking on Nucleotide BLAST from the home page. We see all 5 types of Web BLAST options available here as marked in red. Here we see a form which has 3 sections. First section is Enter Query Sequence. Second section is Choose Search Set. And the third section is Program Selection. In the text box, we can either copy-paste a nucleotide query sequence, or we can type accession number like NM_00032.1.2 typed by me. Alternatively, if you are on an annotation page from nucleotide database, you can click on Run BLAST feature from the links on right hand side. You may also decide to just use a segment of the query sequence in BLAST, searching by entering the range in from the From and To boxes provided under Query Subrange to specify the position of the segment. For example, to limit your matches to the region from 60th base to 180th base of a query sequence, we can enter 60 in the From field and 180 in the To field. If you have saved query sequence in a file, you can also upload the file. You may wish to assign a job title. Additionally, if you want only to compare only your query sequence with another sequence which is a subject, but not use any database sequences, You can tick the box in front of two or more sequences. Now let's enter one or more queries in the top box and one or more subject sequence in the lower text box. Then use the BLAST button at the bottom of the page to align your sequences. Now we move to next section, which is Choose Search Set. This includes a list of databases. Default standard databases are NRNT, which is non-redundant nucleotide, used for BLASTn. Next, we may like to choose a particular organism only to be included or excluded from our search. If not, then we leave this option as per default. BLAST search can also be limited to the result of a query against the database chosen. For example, by typing RNA polymerase not E. coli in organism or entry query search, BLAST will limit a search to all RNA polymerases except those found in E. coli. In program selection for BLASTn, we have three choices, and by default, highly similar sequences or megablast is chosen. Megablast is intended for comparing a query to closely related sequences only and works best if the target percent identity is 95% or more. The second option is for more dissimilar sequences using discontiguous megablast. For somewhat similar sequences but not highly similar, like two organisms may have a distant relationship, the option used is BLASTn. There are some additional algorithm parameters which can be either left as default options or can be reset depending on the requirement of the research question we are having. They help the BLAST to calculate the scores for the query sequence matchings with database sequences. We are all set to BLAST our query sequence now, so we hit the BLAST button at the bottom of the form. It may take a few seconds to a few minutes, so please be patient. Here are the results. Let's learn them step by step. The BLAST result page contains the summary, description table, graphic summary, alignments, and taxonomy sections. The summary section, as marked, is at the top of the result page. This section displays what you searched with, like query ID, etc., and the database you searched against. On the top, we have three options: Edit Search, Save Search, and Search Summary. Edit Search will take you back to the search page. You can rerun your search with adjusted settings. Save Search saves the search settings and you can use them later. Search Summary displays a table with summary statistics and search settings. Filter Results on right-hand side allows the input of organism name, which you can select from the list. This action will filter the returned hits only for that organism. For example, you may want all the data displayed is from humans or Homo sapiens only. Or you may exclude or add any organism from display by checking the Exclude or Add Organism checkbox. Distance tree of results presents the matches in a distance tree format. For phylogenetic analysis, use the Multiple Sequence Alignment or MSA viewer. After summary, the Description tab displays a table of hits found by BLAST. It contains maximum 100 results by default for sequences producing significant alignments. There are three sets of pull-down menus next to it. Download menu helps to download the selected hits, which are those results for which you have checked the boxes to select them. Manage Columns menu has a list of all available columns of the table. You can check or uncheck them to customize your table display. For example, you don't want to see total score column, then you can uncheck this box. Show menu allows you to select how many hits are shown in the table, 100, 50 or 10. By default is chosen as 100. The descriptions table has 8 columns by default. The first column is description. Here we see a description of all the displayed matches in the form of a link as indicated by this arrow, and clicking on them will display the alignment of subject sequence from database to the query sequence which you input. You may choose to check multiple results by checking their boxes. Next column is max scores. BLAST calculates a score after aligning the query and the subject sequence using a scoring matrix. All matching bases are given a positive score, whereas non-matching ones as a negative score. BLAST also adjusts the alignments for any gaps, and gap penalty scores are applied for the gaps. Finally, the total is calculated as max score. Next column is total score. Since BLAST is a local alignment tool, it can align multiple matching regions of the query. In this case, all scores are added to be displayed as total score. If there is no other matched region available, the max score and the total scores will be the same values. Next column is query cover. This tells us whether BLAST used entire query sequence, which will be 100%, to align with the subject sequence or less than 100%. Next column displays e-values or expect values. The expect value is a statistical parameter that describes the number of hits one can expect to see by chance when searching a database of a particular size. It decreases exponentially as the score of the match increases. The lower the e-value or the closer it is to 0 or 0, the more significant the match is. Person identity allows us to know degree of identity between the query and the subject sequences. You may also use filters to sort sequences displaying a particular percentage identity, such as you want to choose between 94.74 and 94.76% identity. The next column, ACC length, allows us to sort results by accession length. The last column displays accession numbers of the most matching sequences, which is subject, sequences as links. By clicking on those links, you will be accessing the annotation page of that particular sequence in the database of nucleotide or protein. Once you choose one or more sequences from the BLAST result table by checking the boxes in front of them, the links for GenBank, graphics, and distance tree of results get activated. GenBank link works in a similar manner as of accession column, whereas graphic links you to a graphical sequence viewer-based presentation of matches anchored by the query. Distance tree of results will display a phylogenetic tree for that sequence in relation to other sequences. Here is a small tree with 4 sequences we chose. The next tab on BLAST results page is the graphic summary. You will see that all the boxes are selected by default. You may choose to unselect all and then select the ones which you wish to check for the graphic summary. This page has a color key based on alignment scores to display results. Scores equal to or more than 200 are displayed as red, scores between 80 to 200 as pink, scores from 50 to 80 as green, scores from 40 to 50 as blue, and scores below 40 are shown as black bars. If you hover on any bar, it displays its title, and if you click, it shows you the pairwise alignment for that sequence. You must have noticed that all the bars are not of the same length. This shows the query cover used for BLAST search was variable. The alignment section contains the detailed pairwise alignment between query and database sequences. Alignments help us to find the matches and mismatches between these two sequences. The gap scores are deducted from the match scores to achieve max scores. GenBank link in the alignment links to nucleotide database, graphics, a graphical sequence viewer based display of the alignment between this database sequence and the query, anchored by that database sequence. This picture shows exons in RB and their locations on the gene. Alignment display includes a mini tab at the top which contains the alignment statistics as shown. Related information links have been given at the right, like a link for gene database, genome data viewer, etc. These links can be used to move to other resources with additional information derived from the matched database sequence. We can download sequences of those chosen results in FASTA format or GenBank format or as alignment or text. Here we see sequences in FASTA format on a notepad which can be saved. Taxonomy tab gets us the classification details of the organism whose sequences have been chosen by BLAST for result display. So friends, this was our tutorial on understanding BLAST as a beginner. Hope you enjoyed and understood. In our next video, we will learn about PubMed database. If you are enjoying these tutorials, please like and subscribe to my channel and keep me going for the next series. Bye bye and see you soon. Thank you.